Before you can learn, memorize, or study anything, you need to perceive it. Sounds simple, but this process involves a number of biological processes that many psychology textbooks often ignore and yet are fundamental to understanding the field, not to mention fun to look at. To understand cognition and certainly to assess the cognitive psychology models that we will describe throughout this series, you need to know what information the brain receives. For this reason, studying perception is vital for understanding cognitive psychology. Although we have all sorts of senses, in this video we will explore the eyes and how the brain uses the information they provide. I will establish that seeing is a highly psychological process and affected by cognition. Furthermore, I will demonstrate that seeing isn't always believing by filling you in on some cool visual illusions that highlight how easily the human visual system can be tricked. Such illusions reveal important information about how people see and why they see what they do. So, everyone has the same basic biological setup in order to sense the world: eyes, ears, nose, and so on. But people don't see and hear things in exactly the same way. This is due to the psychological processes involved in such perception. Now, two early schools of thought in psychology made the following important distinction between sensation and perception. They said that sensation is how the senses and the brain detects and transmit information about the world. Perception, on the other hand, is what person's consciousness sees and responds to. Um, Much of human behavior is actually responding to the environment. And so, psychophysics was devised to explore exactly how this occurs. In other words, to explore the limits of human perception. Now, the main proponents of psychophysics research was Gustav Fechner, a German physicist, psychologist, and philosopher. His logic was to find the minimum sensation to see a particular stimulus or to distinguish between two stimuli. The psychophysics approach treated humans as physical organisms and ignored things such as free will. Now, psychophysicists weren't interested in how people interpret what they see. They just that they did. Therefore, it's very objective and based solely on data. Now, an alternative school of thought primarily interested in the quality of people's perception was structuralism, attributed mostly to Wilhelm Wundt, a German physician, physiologist, and philosopher. Now, Wundt suggested that the mind represents the world as individual elements with many different cognitive processes that interact with each other. The mind takes what the body senses and creatively attempts to understand it based on internal structure. Now, structuralists thought that the best way to research the human experience was analytic introspection. This approach requires a detailed verbal description of what's being seen or thought about and why this happened and why this is happening. The data produced a highly subjective based entirely on individuals' own opinions. Psychophysics rather is still widely used today, whereas structuralism is rarely taken seriously. Now, your brain sees the world differently to someone else's, and possibly in a way that isn't exactly correct, because it's always trying to make sense of the world in order to respond to it in the best practical way. Sensing and perceiving the world is vitally important. you are constantly receiving sensory stimulation. Without such sensory information constantly streaming into your brain, you may not even be considered alive. If you're in a sensory deprivation chamber, a device used for meditation purposes, your brain creates sensations in the form of hallucinations. Without stimulation, without stimulation, people's brains create their own sensation. Perception, on the other hand, is critical to action. In order to do something, you need to perceive it first. When you see a chocolate bar, you need to use all the information your eyes give you to reach out and pick it up. Then you use the combination of information from the eyes and fingers to open it so that you get the pleasure of the taste. People use many different senses to understand the world, as well as vision, hearing, touch, taste, and smell. You can also detect much more, such as balance and temperature. Although in this video we look only at vision, note that each sense gives you different information with very little redundancy among them. Information is generally only produced by one of the senses. That's not to say that senses are entirely separable. However, vision can affect tastes. For example, vision can affect taste. For example, now the eye is one of the most amazing organs in the human body. It allows you to see in so many situations, from the bright sunlight on the deck of a cruise ship to a dimly lit nightclub. Even in these two widely different environments, your vision is sufficient for you to move around safely. As these examples suggest, the crucial element in seeing is light. It carries the information that you use to see the world and is extremely useful for sensation. Light interacts with many things that it touches, reflecting off some objects and being absorbed by others. And in this way, providing information about what objects are. Fortunately, the world has lots of light. The sun, note that the sun produces a huge amount of it. Now, the eye is the device that detects the light carrying the information you need. For example, to perceive objects. Have you ever wondered why your eyes are placed where they are? Well, wonder no more. Certain animals have eyes on the sides of their heads and others have them on the front for very good reasons. Animals with two eyes facing forward have greater visual abilities for things in front of them because light enters both eyes. Therefore, chasing other animals is easier for them, though they can't see much at right angles or more to the sides of the head. Animals with eyes on the sides of their heads, such as rabbits, are able to see almost all the way around their bodies, with the exception of the very back part. They're much less able to see fine details in front of them. However, these animals are often prey and need to be on constant alert for animals that hunt them. The eye is a water-filled jelly-like ball with a black hole. It's really just a hole at the front. This is called the pupil to allow light in. Behind the pupil is a lens, like those in specs like this, designed to focus the light coming in on the back of the eye, which is called the retina, where the photoreceptors detect the light. To let more light in when you move into the dark, the pupils widen. To reduce the amount of light coming in the eye and prevent damage to the delicate parts of the eye beyond, the pupil constricts. Of course, pupil width can change in response to psychological factors too, such as when you see someone you like. Now, the photoreceptors are at the back of the eye, behind layers of other stuff. They're distributed all over the retina, but with many more cones in the fovea, which is the middle of vision, than in the periphery or the edge of vision. Cones are photoreceptors that respond in bright light and are good at color detection. Rods are photoreceptors that respond in dim light and aren't great at color detection, which is why detecting color at night is harder when you compared when you are trying to detect color at daytime. Now, this uneven distribution of photoreceptors means that the fovea is the place of maximum resolution. Seeing fine detail is much easier or has higher visual or because you have higher visual acuity in the fovea, and the periphery appears more blurry, which is why people move their eyes to look at important scenes. Now, the eye muscles are the busiest muscles in the human body. They move more than 100,000 times in a given day. In fact, eye movements are so important that without them, you'd go blind. You don't believe me? Why not check this out? This is called the Troxler fading. And what I want you to do is stare at the plus sign in the center and watch as the shading around the outside begins to fade away. Now, this effect happens because your brain works by comparing one retinal image with the next. With no difference, the brain thinks that nothing is present. The brain sees by detecting differences between two scenes, and if you paralyze the eye muscles, you see nothing at all. You require your eye muscles to see even though they're not involved in the detection of light. Now, in 1962, Nobel Prize winner neurophysiologist David Hubel and Torsten Wiesel discovered the interesting layout of visual cells beyond the retina, which are called retinal ganglion cells. By shining a small bit of light onto each cell and measuring the response that these cells gave, they discovered what cells respond to in the receptive field. They found that the cells have a donut-shaped layout called the center surround layout. Now, the center responds to light being on, whereas the surround responds to light being off. You can have the opposite layout. You can have the opposite layout too. This layout ensures that you can see changes in light much more easily. It also helps you to see small things. In a small if a small bit of light shines on the center of an on-center cell but doesn't touch the outer part, the cell responds a lot. However, if light shines across the whole cell, both on center and the off surrounding parts, the cell doesn't fire. Now, light moving across a cell causes a small cellular response, except for the brief period when only the on center has light on it. One of the consequences of the center surround layout is that sometimes you see things that aren't there. Consider the Hermann grid. So you should see illusory gray spots appearing in most of the white intersections. Now, this effect occurs because at the intersection, more light is on the off surrounding than the on center of the cell's receptive field, causing the whole region to appear to have no light. The effect doesn't occur in the fovea because the cells here respond to a smaller region. If you bring your phone or tablet closer to your head, which you are if you bring your phone or tablet which you are using to watch this video sufficiently close to your fovea, you get illusory dots in the intersection you are looking at as well. Now, um, this example demonstrates that vision is an active process and that the brain changes and distorts what you see. As a result, detecting whether you have visual problems is difficult because your brain always tries to compensate. Now, this filling in is incredibly important. If you're uh, if you're crossing the road, a car can be in the blind spot and you don't see it. So the road safety advice is right. Always look. Look away and then look back again. Now, the information from each eye leaves via an optic nerve. Damages to the optic nerve means that you lose vision in one eye because at a point the optic chiasm uh, information from the two eyes mixes. Information representing the left of what's seen in each eye is seen in the right optic tract, and information from the right on what's seen is sent to the left optic tract. If one optic tract is cut, you lose vision for one side of the world called hemianopia. So, cells from the optic tract go to the part of the brain called lateral geniculate nuclei or nucleus or LGN for short, which is arranged into 6 layers. 2 layers are magnocellular and are sensitive to movement, and the other 4 parvocellular layers are sensitive to color and show high visual acuity. Now, the neurons take visual information from the LGN to the primary visual cortex or area V1 of the brain in the occipital lobe. Now, this is the first area of the brain that generates conscious visual experiences and is located at the very back of your head. You can tell that this is the center for vision because if you stimulate cells using brain stimulation or a hard strike in that part of your head, you see something. If you hit the back of the head hard enough, you see stars like that in cartoons. And this response comes from neural activity in the visual cortex, but please don't try this at home. Now, the primary visual cortex is said to be retinotypically mapped. It's like a map of the visual world. Something that's next to something in space is processed by cells next to each other in the brain. Now, some cells in the primary visual cortex are simple cells, the basic building blocks of vision. They detect and process simple shapes such as edges or bars, but each cell processes only the edges in a particular orientation. A vertical bar or a detector doesn't see horizontal bars. Simple cells exist for many different types of edges, bars, and lines, with more cells for more frequently encountered edges. Humans have more cells for horizontal and vertical bars than for oblique angles. Cats have cells for all orientation. Perhaps that's why they often look at humans with superior disdain. So, humans find detecting horizontal and vertical lines easier than those of other angles, the oblique effect. Now, in other words, they have greater visual acuity for horizontal and vertical lines. Now, the oblique effect shows how experience affect the cells in the brain. For instance, people living in the rainforests or the North American plains don't display the same oblique effect. Those in the rainforests have fewer horizontal cells, and those in the plains have fewer vertical cells. So, in the brain, the response of several simple cells can be combined into a complex cell. A complex cell can process a vertical bar, but of any size but of any size and anywhere in the visual world. Farther into the brain, the responses of several complex cells can combine to form what is called hypercomplex cells, which responds to even more complicated sets of patterns, such as a moving vertical bar. Um, by this simple process of adding up the responses of the cells, people create the entire world. If you combine a series of simple cells representing curves of particular orientation, you can construct a hyper complex cells for faces. Now, given this knowledge, perhaps a hyper complex cell represents everything that you have seen, adding together the activation of many simple and complex cells representing the edge that make up the object. So, you'd have a cell for every single thing that you have seen. Now, this is called the grandmother cell argument. To see your grandmother, you need a cell that represents your grandmother. Indeed, brain imaging research shows cells that seem to respond only to Jennifer Aniston, which seem to indicate that the grandmother cell argument is valid. But if this were the case, people would need many more neurons in the brain that they already have. To address this major flaw in the argument, instead of each cell responding to a single thing, maybe a collection of cells together respond, and some of these cells overlap with other objects. Now, the primary visual cortex or area V1 is associated with limited and basic visual processing. More detailed processing occurs later in later brain areas. However, two main visual pathways appear to exist after V1. The what pathway, which is designed to establish the identity of what you are seeing, it's it's described as the temporal stream because it involves brain regions in the temporal lobes that process memory. It's also called the dorsal stream because it rises over the top of the head like a dorsal fin of a whale. Now, the crucial brain regions involved in the what pathways are areas such as V4 and V3, which processes color and shape respectively. Now, this stream is considered vision for perception. Now, the where pathways designed to establish where something is, it's more basic than the what pathway. It simply signals the location and movement of something but not what it is. This pathway is described as the parietal stream because it goes to the parietal lobe, and sometimes the ventral stream because it goes low through the base of the brain, closer to the stomach. Ventral is actually stomach in Latin. So it passes through area V5, which processes movement. Now, this stream is considered vision for action. Now, the idea that the brain breaks up the world into many separate properties such as color and form, known as functional specialization, is the subject of extensive debate among psychologists. After the information is broken up, higher brain areas must then bind all the information together to form a complete representation. So here's another interesting thing when it comes to looking at your eyes or your visual system. We all have a blind spot, or what is called a scotoma. It's an obscuration of the visual field, or a particular blind spot known as the physiological blind spot, or blind point is the place in the visual field that corresponds to the lack of light detecting photoreceptors on the optic disc of the retina, where the optic nerve passes through the optic disc. So, in order to identify your blind spot, what I want you to do is to close your right eye. No, your left eye, that is. So, I want you to close your left eye and look at the cross in this image. So, you can either bring your phone or your tablet closer to your eye or closer to your face, or bring it farther away to your face, and at some point you will realize that the letter A on the other side of the line disappears. Now, if you're using a laptop or a computer that is, well, difficult to bring up, to bring closer to your head, maybe what you can do is to move your head closer to the screen, or adjust your head farther to your screen, and at some point the letter A on the far side of the line will begin to disappear. However, as soon as you try to look at the letter A, it will then reappear. This is called a blind spot, or a scotoma. So, another example of how what you think you see isn't necessarily exactly truthful comes from After Effects. This is where the world appears different following prolonged exposure to a particular stimulus. So what I want you to do is to stare at the fixation point between the gratings on the left for about a minute. Then, I want you to look to the fixation point between the gratings on the right. You should see an after effect where the top gratings seem narrower than the bottom gratings. Wait a while and look back at the gratings on the right. You can see that in reality, they're the same size. So, this exercise changes what you can perceive by adaptation, or prolonged exposure to a particular stimulus affects the ability to see the stimulus later. Now, looking at the gratings close together, which is considered with high spatial frequency, fatigue fatigues or tires out the cells in the brain that responds to them. Then, when you look at other gratings, these cells no longer fire, and so you see the world without these spatial frequencies. This example highlights how perception is constructed by your existing knowledge. Now, American psychologist James Gibson believed that perception was designed to respond to the visual world. This direct perception approach is similar to the idea that the sole purpose of perception is to help initiate some form of action. Gibson believed that when people encounter objects, the way the objects are used is immediately brought to the mind, so objects' uses are their affordances. So, when someone encounters, let's say, an apple, he perceives the process of eating. However, his psychological state affects the affordances. So, if he is angry, he may perceive the affordance of using the apple as a projectile weapon and throw it. Gibson's direct perception theory from the preceding section of this video predicts that knowledge is available instantly when you perceive the world. Now, the knowledge is about how an object may be used. But some objects have no use, and yet knowledge is known to affect the perception. One example of this is what is called the ambiguous illusion. So, I want you to look at this image and, well, look at it and tell me what you can see. So, people see either an old woman or a young woman. Well, in fact, the perception of the two can change in front of your eyes, despite the fact that the visual input hasn't altered or hasn't been changed. This effect means that the brain must be involved at the first stage of visual processing. If you can still if you can't see if you can still see the old or young woman, the young woman is facing to the left, looking upward. Her chin is the old woman's nose, because she's looking down to the left. Now, illusions such as this one show that knowledge and context affect greatly what people see. In fact, knowledge and experience have quite amazing effects on perception. The more experience you have looking at something, the less you need to look at it to see it. Plus, experience of particular objects speed up the brain's processing of them. For example, expert chess players can look at a chessboard and see the entire board with one glance at the center. Whereas novices like me need to look at each piece to have the same representation. Imagining a world without movement is almost impossible. Movement is one of the definitions of life, and surely is so perceiving movement. Motion perception is fundamentally important for survival. Imagine trying to cross a road without it. Plus, many other less obvious purposes lie behind your ability to perceive motion. Now, being able to perceive motion helps people to detect things. If you're hunting your food in a forest, as we surely do most days, the slightest motions attract your attention and help you get your next meal, as well as help you to avoid becoming somebody else's meals. Now, in this part of the lesson, I will describe the intricate brain processes involved in detecting motion, including the involvement of optic flow, and then I will demonstrate the importance of motion detection of timing, discuss the special role of biological motion, and show how your motion perception can easily be fooled. So, the question of how people perceive motion isn't as simple as it sounds. Consider these two distinct cases. We have what is called retinal movement system. When you keep your eyes still and something moves in front of them. In 1 second, you see the object in one point in your retina. In the next instant, the object is in another position. How do you know it isn't two objects? Well, another is that the eye, or rather the head slash eye movement system. When you move your eyes to keep an object in your fovea, in other words, you track the object using smooth pursuit eye movements. So a third case exists where you're moving and you have to move your eyes to keep the object still in your vision. But here, you don't perceive things moving because you realize that you're moving yourself. Now, scientists have devised two theories to explain how people achieve the incredible feat of motion detection. You have inflow theory, where Charles Scott Sherrington, a British neuroscientist, suggested that the brain keeps a record of how much people move their eyes rather. Now, that is, the brain tells the eyes or head to move and then records how much movements happened. This information is compared to the changes in the retinal image. Now, the second theory, otherwise known as the outflow theory, where Hermann Ludwig Ferdinand von Helmholtz, a German physicist, suggested a subtly different approach. Now, in this theory, the brain plans to move the eyes, creating an efferent signal, which is a signal representing this plan. Now, the brain compares this plan to what the retinal image displays. So, how do these two theories cope with what has been observed in the real world? Both theories can easily explain smooth tracking, which is following an object smoothly with your eyes. Though no retinal motion occurs, both theories record that the object is moving because the eye muscles are working well, which is called inflow, or the brain tells the eye muscles to work, or the outflow. But if you poke your eye, which I don't want you to do, you see movements too. Uh, j- Please don't jab your finger in your eye right now. Take, as much as possible, just take my word for it. So, the muscles have moved, but the brain didn't tell them to move. So, the world should remain stationary according to inflow theory, because the muscles have moved and the brain should register the same thing as tracking. Yet, you perceive motion when you do this. Again, please don't do this. Just take my word for it. So, outflow theory suggests that the brain made no intention to move. And so, because you saw motion, the eyes have moved, which is what happened. Based on this and other evidences, outflow theory is more valid in explaining how people detect motion. But although it shows why your brain may perceive motion, it does not explain how the cells in your retina respond to it. to understand this aspect, you need some form of motion detection system. So, a motion detector works by comparing the signals from two different receptors. It requires two receptive fields. Now, a comparator cell and a time delay, which are needed in this system. So, a stimulus is caught on receptor A and then moves to receptor B. Now, the comparator cell activates only if the activation at receptor B is delayed after the activation at receptor A by a short amount. So, this model is known as the delay and compare detector or Rayshard detector. So, speed detection is possible by having comparator cells that respond to different levels of delay. A shorter delay means that something is moving faster. A longer delay means that something is moving much slower. Evidence suggests that the motion detection is occurring in area V5, otherwise known as MT, in the visual cortex. Now, cells in this part of the brain process motion in every direction. That is, each cell responds to motion in a particular direction. When you walk around, you often focus on a particular point. Well, this is your intended destination. This focus of expansion appears motionless. Now, the visual field around that point, however, appears to be expanding around it, or, well, getting bigger. This pattern occurs during optic flow, which is highly useful in determining direction of travel. On average, the error in predicting heading in predicted heading based on optic flow is very small. Although optic flow is useful, how it works it isn't entirely clear. Now, in 1950s, James Gibson, the dude that we've talked about earlier, who well, introduced the direct approach to perception, suggested that people somehow use their entire visual field to make a judgment of direction from optic flow. So, um, in contrast, another theory says that you need only to focus on a single location element in the visual field that's normally stationary. Evidence points to the fact that judgments of direction can be made based on optic flow when no single stationary object exists, suggesting that the local theory is less valid than Gibson's global theory. When you're in a train looking out the window and the train in the platform next starts to pull out, often you can think that your train is the one moving, possibly out of a forlorn hope that your train will actually get you home on time. Now, this effect is an example of something called induced motion, but more specifically, vection or self-motion. Now, one method for showing vection is using an optokinetic drum or a rotating room. Participants are placed inside this rotating room and then they start to think the room is rotating. But for some reason, they mistakenly believe they're the ones rotating and not the room. Now, this reason is that the vestibular system, which balances signals, or which signals balance, only signals changes. You're inside, or you're used to receiving a signal only at the beginning of the motion. So, if you see the room move but you don't get a signal, your brain is confused but perceives that the room is moving. After a few seconds, your brain is no longer confused. So, because it's receiving no input from the vestibular system, and so the room can't be moving. But you can be. So, Vection has been linked to causing vomiting because of the mismatch between what you're seeing and what you're actually going through. So in 1976, British psychologists David Lee and J.R. Leishman used vection to show how movements of the environment can affect balance. They put toddlers who had just learned to stand up in a room and waited until they stood. They then moved the walls slightly. The effect caused the toddlers to fall over. If they moved the wall in front of a toddler, the toddler fell backward. The toddlers mistakenly assumed that the walls was moving because they were moving too. And so they learned to compensate for it. You know what else use vection? IMAX cinemas. So, motion helps people to extract three-dimension information about an object. If it moves, its size changes on your retina and you can perceive its distance, known as obtaining structure from motion. An example is when you can interpret a static image in a number of ways. Say that you can see a shadow of a straight line. Now, the object casting the said shadow could either be a piece of string or a hard stick. If the object suddenly moves or is moved, you can now gain information regarding its structure. You'd see a stick move in rigid ways, whereas a string would bounce in the wind. So, a special case of obtaining structure from motion is the ability to obtain information about biological motion. Movement from a living creature. So, it can be distinguished from mechanical motion, which is rigid and harsh. You know that if you have seen robots dance on YouTube videos. Now, biological motion is much more fluid. Biological motion is very important for humans to detect, providing them with information about predators or dangers or prey. So, this importance means that humans are expert at reading biological motion. People can often recognize their close friends simply from the way they walk. Biological motion can also provide information about someone's age, gender, and even sexuality. So, the expert nature of biological motion is highlighted by the fact that if you reduce the amount of information to the bare minimum, people can still recognize biological motion. In 1975, Gunnar Johansson, a Swedish psychophysicist, used points of light attached to the joints of some people. Sort of like when you see game producers uh, copy the movement of let's say actual basketball players to make the images or the movements of in-game character to appear more fluid. So he then asked them to move in particular ways. Now the people were recorded wearing all black on a black background, leaving only the point light motion visible. Now without motion, people couldn't identify the point light object stimulus. But as soon as the person moved, the participants easily identified the stimulus. In fact, people can readily recognize different types of movement from point light displays. Painting, doing push-ups, riding a bike, and they can lip read too. So, participants can even recognize other people's gender from point light displays. So, they can identify their friends, family members, and even dogs and cats with minimal information. Furthermore, they can extract this information very quickly from point light motion. Participants can make judgments in less than 200 milliseconds, suggesting that the process is fairly automatic. Humans aren't the only creatures able to do this, however. Cats can identify other cats from point light display. Actually, point light displays are used widely in both films and video games. Many computer generated characters such as Gollum here or the Hulk in MCU in MCU in MCU movies, in Marvel movies, were filmed with point lights. So the technique enables the computer animators to ensure that the computer generated character moves in a realistic manner. Now, biological information is so important that specialized brain regions may be devoted to processing it. Now, researchers have identified clusters of cells in the superior temporal sulcus that seem to be specially recruited when processing biological motion. Lastly, we have to talk about apparent motion. Now, the way cinema displays film is similar to the way in which human visual system encodes movement. Now, in the cinemas, a series of still images are presented at precisely 24 images per second. Now, the image of each frame means that audiences perceive a smooth apparent motion. This may seem strange, but this may seem strange because the human visual system can detect flickering at this speed. But film projectors can, or rather, have shutters that open up to 3 times per frame, creating a flicker rate the visual system can't detect, nearly 75 frames per second. Images are presented at this rate because if the frames were presented much more slower, you wouldn't perceive motion. Just a series of still frames. If the frames were presented at a much greater rate, you wouldn't see motion but everything simultaneously. So, if the image appear at receptor B sooner than the delay from receptor A allows, the image is perceived as happening at the same time. If the image appears at receptor B much longer after the delay, you perceive seeing two separate still images. Thus, the frame rate that people detects as motion is the same as the delay in their motion detectors. So another form of apparent motion is quite fascinating. If you look at the hubcap, or otherwise known as mags of certain cars when in motion, it appears to be rotating either to the direction of the car's movement or in the opposite direction to the motion of the car, often called the wagon wheel effect. Now, when the wheel is moving slowly, your motion detectors detect frame one because the spokes are in a one position. Or rather at frame 2, however, the spokes have moved a little and your motion detectors assume they've moved forward. But if the wheel is moving too quickly, by the time frame 2 happens, the spokes have moved so far around more than 45 degrees that you think the wheel is moving in the opposite direction. So, in this video, we differentiated sensation and perception. We also looked into the physiology of sight and discussed the psychology behind vision. Again, my name is Dex Camitan and thank you for watching.